Hello and welcome everyone to this podcast. In this podcast we're going to talk about the cytoskeleton. In fact, in the next several podcasts we'll talk about the cytoskeleton. Today I'm going to talk about a brief introduction to the cytoskeleton, and then I'm going to focus on the first type of the cytoskeleton called Intermediate Filaments. So let's go ahead and get started. The cytoskeleton in eukaryotic cells is important for several reasons. It's important in maintaining shape, movement, and when I say movement, I mean both intracellularly, meaning to move things within the cell, and extracellularly, meaning moving that cell from one place to the next. They're important in cell division, muscle contraction, interacting with other cells, and many other things. Now, when we think of the cytoskeleton, we can think of three types of cytoskeleton. The first of which we're going to talk about in a little bit are intermediate filaments. The second are microtubules. And the last one are actin filaments. Sometimes actin filaments are called microfilaments. The subunit of intermediate filaments are intermediate filament proteins. They have several functions, and we'll talk about a few of them, but for now let's just think of them as giving the cells mechanical strength. For microtubules, their subunits are the protein called tubulin. So microtubules are made up of repeating units of tubulin. One of their biggest functions is involved in moving vesicles, organelles, DNA in a cell. Actin filaments or microfilaments, their functional subunits are the protein called, this should be easy to remember, actin. Like the other two, they have many functions, but for now let's just think of them as providing structure to the cell at the cell cortex. So that's the part of the cell right below the plasma membrane, and we'll see these actin bundles near the cell cortex providing that cellular structure. Okay, now let's move on with a little bit more in-depth discussion of intermediate filaments. And I'm going to begin with just some characteristics of intermediate filaments. These are the essential things that you should know about intermediate filaments. As I said on the previous board, their main role is protecting cells against mechanical stress. So when that cell is stretched, it protects that cell from breaking apart. These intermediate filaments do. They're called intermediate filaments because the first intermediate filaments identified had a size in between actin and microtubule size. Not surprising based on the role of the three kinds of cytoskeletons. Intermediate filaments are the strongest. And the way they showed this first was that adding salt to the intermediate filaments did not disrupt them. Adding salt to actin or microtubules did disrupt them. So these are the strongest of the three. Intermediate filaments are well distributed in a cell. So they're found throughout the cytoplasm, and more specifically, you will find them surrounding the nucleus. They are often found anchored to the cell wall by this protein structure that we'll talk about later called desmosomes. We'll talk about these towards the end of the semester, but these are structures that hold cells together. And from here, they will interact with other intermediate filaments across the cell. And in some ways, then, you can think of this as how they can interact and provide that coordinated strength between cells because they are interacting with each other through these desmosomes. Let me also draw the nucleus here, because if you remember above I said that they will often find themselves surrounding the nucleus. They are also found in the nucleus. And in the nucleus, they are usually found on this very inside of the nucleus, where they form, and we'll talk about this in a little bit, but they form this structure called the nuclear lamina. And we'll come back to that in a little bit. As you can see, the intermediate filaments have a very wide distribution throughout the cell, more so than we will see with microtubules or actin filaments. Now let's talk about this structure here. How do we form this really strong structure? We often think of the structure, so the intermediate, I'm just going to call them IFs, the intermediate filament structures, to be rope-like. So if you remember, I said that the subunits of these are these intermediate filament proteins. Those are the subunits. And what I mean by that is we have right here just one large protein, long protein, has the N terminus here and the C terminus here. It will form a dimer with another one in the same orientation, N terminal to C terminal, by forming this helix. So in looking at this dimer, I want you to keep in mind that we have N terminal on both of the subunits on one side and the C terminus of the subunits on the other side. That's important because what happens next is we will form a tetramer containing 4 subunits. And this other subunit will be in the reverse orientation. So I'll do my best to draw this as a tetramer, but the key is we've combined 2 dimers in opposite orientations. And they're staggered a little bit. So we say this tetramer here is staggered and anti-parallel. And then what happens is these tetramers, what will happen is that these tetramers, shown down here on the bottom, will bind to other tetramers. And what results then, I'm going to draw it up here, but I'm going to erase what we see here, is we will see repeating units So when they come together, these tetramers, you might see something like this, where this tetramer and these tetramers here all kind of interact with each other, both in a horizontal way and a vertical way, much in the same way that a rope is formed by the strings, smaller strings, wrapping around each other. By forming this very highly ordered structure here, it provides that strength that's needed as that cell stretches or bends to allow the cell to survive and withstand that mechanical stress. Now what I would like to do is talk more specifically about these filaments and the different parts of the filament. So let's look at a single filament and talk about the parts of it. So this is just drawing it N terminus to C terminus, and I'm going to make it 2 different colors here, and I'll explain why in a moment. But it's still the same protein, the same filament. Here we have the invariable region. Over here we have the variable regions. All of the types of intermediate filaments there are, and there are several, there's at least 6, and they will all have this same invariable region, which helps in the binding of neighboring intermediate filaments to provide that helical structure and that advanced structure. And the part that varies is the part over here, and I'm just going to draw this in a different color. Still an intermediate filament, but they may be of different sizes. They will certainly be of different sequence, even if they happen to be the same size. But by having this variable region, we're allowing that intermediate filament to have different functions. And we will see that intermediate filaments have variable functions in different cells. Within the neuron, they have a different function. Within glial cells, they have different functions. Within muscle cells, they have different functions. And that functional difference between the intermediate filaments is all because of these variable regions. What I'd like to do now is to talk briefly about 4 different classes of intermediate filaments. The first are keratin filaments. These are found largely in epithelial cells. Epithelial cells include the lining of our GI organs, like our stomach, our intestines, our tongue, the cornea in our eye. This is what our hair is largely made of, or if you happen to have hair, it's largely made of that. Feathers, for those of us who happen to be birds, those are keratin filaments. And we'll talk a little bit more about those in just a moment. The second type of intermediate filament are vimentin. These are found in connective tissues. This would include muscle and glial cells. Glial cells are the cells that support the nervous system, like astrocytes would be an example. The next one would be neurofilaments. And these, as you might guess, are found in neurons. And the next one are nuclear lamins, which we've briefly talked about because they make up the nuclear laminin. So these are going to be found in the nuclear envelope, associated with the nuclear envelope on the inside of the nucleus. So these first three, even though I didn't specifically say it, these are all in the cytosol, whereas the nuclear lamins are in the nucleus. I'm going to spend just a little bit more time talking about keratins. Keratins are really important in providing that tensile strength that'll protect the cell against mechanical stress. Part of the way they do that is binding to these desmosomes. We talked about them before, but let me go ahead and label them again. These are cellular junction proteins that will interact with neighboring cells to hold cells together. So let's go ahead and draw these, but remember, these are going to be that highly structured, rope-like structure of these intermediate filaments. And it's not just one across here, they are going to crisscross the cell in many different ways to provide that strength throughout the cell. But by being connected to these desmosomes that are connecting the cells, it allows the intermediates of neighboring cells to be indirectly connected to each other. Because I should actually draw these desmosomes in such a way that they are connecting the cells directly. So the actions of the keratin in this middle cell impacts the cells next to it because of this connection through the desmosomes. And that helps provide this tensile strength and protecting against mechanical stress as you begin to pull these cells and stretch these cells within the tissue. I want to mention one protein that we haven't talked about yet, and these are called lectins. Lectins allow intermediate filaments to form bundles, but also to provide the link or connection, might be better to say, between intermediate filaments and microtubules, or intermediate filaments and actin. So we may have actin interacting with the intermediate filament through these lectin proteins, or we may have microtubules interacting with the intermediate filaments through the lectin protein. Now I want to spend some time talking about nuclear intermediate filaments, the ones that are inside of the nucleus. And as I mentioned before, these are called lamins. Structurally speaking, they're different than the cytoplasmic ones. So in the cytoplasm, they form these rope-like structures that I've drawn for you before. In the nucleus, they form this interconnecting mesh work of crisscrossing intermediate filaments. Both the ones in the cytoplasm and the nucleus provide that structure and that protection, but they do it by forming slightly different structures. I'm going to draw just the nucleus here, but you can consider the whole whiteboard if you'd like, the cell. This nuclear lamina, as I said, will form this kind of two-dimensional meshwork around here. So these are lamin proteins that together form the nuclear lamina. So collectively, this orange circle that I've drawn here is the nuclear lamina. But the fundamental proteins that hold it and make it up are the lamin proteins. And there are all kinds of categories of these lamin proteins that are specific to certain kinds of cells and certain kinds of developmental periods, which we're not going to get into. For now, you can just know them as lamin proteins. This nuclear lamina has several important functions, which I'll start listing over here. They provide structure to the nucleus. They also help regulate replication and cell division. And they're incredibly important in transcription. So within this structure here, we will have various chromosomes associated with it, linking directly with the nuclear lamina. And the nuclear lamina holds the chromosomes in a very unique pattern. So let me give this key here. These squiggly lines here were an attempt at a double helix, and these are the chromosomes. Now they don't just hold them randomly, they hold them in very precise ways so that certain parts of this chromosome can be replicated or transcribed. Different parts of these proteins will, these chromosomes will form these interconnecting regions in these cells with these regions within the nucleus that we call transcription factories. It'll rotate and put very specific parts of the chromosome into these transcription factories to turn on genes specifically that are in these regions. And so when this nuclear lamina breaks down, it can have consequences to gene expression. Now, the last thing I want to talk about with these nuclear laminins is that in order for the cell to divide and the nucleus to segregate its chromosomes, these nuclear laminins have to break down. And they do this by a process of phosphorylation, using our good friends, the kinases, and dephosphorylation, using phosphatases. So how do phosphorylation and dephosphorylation cause the nuclear laminin to break apart during cell division? Phosphorylation causes a conformational change that weakens the tetramers, and the nuclear filaments fall apart. Now, at the end of cell division, these places where they've been phosphorylated will be dephosphorylated, and this strengthens the tetramers and their association with the nucleus. Okay, that's all I have to say in this podcast. We introduced the three types of cytoskeletons, and then we focused on the intermediate filaments, their roles, how their structures are formed, and then we talked about specific types of intermediate filaments. If you have any questions at all as you're going over this material, please feel free to come by. If not, I'll see you in class.